Good afternoon, everybody. Welcome to this webinar, which is part of the Biotech Start-up Series organized by BioLabs and Servier. This is a regular conference, a series of conferences and webinars that we organize to gather entrepreneurs in biotech. But before we go into the substance of the webinar, I just would like to mention a few housekeeping points. This talk will be recorded. It will be posted both on BioLabs' website and on Servier's YouTube channel very shortly. So if you want to revisit any part of the meeting, you will be able to do so. The chat function of the Zoom webinar is disabled. You will not be able to use that. You can use the Q&A function at the bottom of your screen to ask questions during the webinar. And we would definitely like to encourage you to ask questions. The staff will be collecting the questions and will be transferring those to the moderators, myself and Olivier from Servier. We do want to keep 15 minutes at the back end of this session for your Q&A. So don't be shy. Do not be disappointed if the staff does not acknowledge your question directly. Know that they will be received and read. And we will try our best to address all of your questions. Don't assume somebody else has asked a question. You will not be able to see the participants' questions. So again, don't be shy and please ask away. Also, after the webinar, we will ask for your feedback. So please kindly fill the quick questionnaire when you exit the Zoom webinar. Your feedback will help us make these conferences better. So we really look forward to that. And as a last point, after the webinar, we are organizing a networking session. You should have received a separate link to another Zoom meeting. This is not going to be a part of the same meeting. You will have to connect into a regular Zoom meeting, not a webinar. And that link is posted in the chat section right now, and we'll post it again at the end of the webinar. The networking itself will last about 20 minutes after the webinar, and we certainly hope you join us at that point. So again, for those who are just joining us right now, the webinar is recorded. Please ask all your questions in the Q&A. Fill the questionnaire at the end, and we will be glad to have you participate in the networking session afterwards. So with this, I would like to introduce myself and introduce my partner, Olivier from Servier. And Olivier, maybe you can have your video on as well. So my name is Jean-Jacques Yarmoff. I work at BioLabs, and BioLabs runs innovation spaces in the United States. Incubators could be another way to describe what we do. But the companies that are members of our innovation communities find much more than shared laboratories. Today, BioLabs has a network of nine shared facilities in the US, and we host about 250 biotech companies in our walls. Now, we're planning to expand to Europe and specifically to France, and we have partnered with Servier. And it is in order to meet the entrepreneur community in Europe that we have launched this series of conferences and webinars, of which this is the second installment. We try to bring to biotech entrepreneurs the tools and the subject that we hope will be useful. Useful to grow your company, useful to understand the changes in our business. But first, let me introduce my friend and partner in this program, Olivier Nogent, who is in charge of the Open Innovation Group at Servier. Olivier?
Thank you, Jean-Jacques, and welcome everybody to this session of our series on startup and biotech conferences. Servier is a natural partner of innovative small companies in health sciences. We have a range of partnerships dealing with either therapeutic innovation or technology innovation that will support our drug discovery and drug development activities. We have an international footprint, being present in 150 countries and being present in the US for a few months, and partnering in the US too with BioLabs and supporting networking and exchanges with companies over there. What we're trying to do with by supporting this series of conferences is to bring to the French environment of entrepreneurs some insights and some knowledge on the hot topics that we have today in health sciences. And I'm very happy to be here today and to enjoy this extremely interesting topic on how artificial intelligence will and is already disrupting our activities. So, Jean-Jacques.
So, thank you, Olivier. And so today we are going to focus on the use of artificial intelligence as it pertains to the early phases of drug development. So without any further ado, I would like to introduce our two speakers, Laurent Audoly from Parthenon Therapeutics and Anna Huygues-Despointes from Owkin. So Anna, if you would please introduce yourself and introduce Owkin as well. Thank you.
Hello everyone, can you hear me well?
Hello Anna, yes.
Okay, so first of all, thank you for having me. It's a pleasure. Obviously we know Servier quite well, but we are glad to do this first collaboration with BioLabs. So my name is Anna Huygues-Despointes. I'm the head of strategy and marketing at Owkin. I've been with the company for 2.5 years. Prior to that, I was an investor in machine learning, AI, in healthcare and agriculture. And prior to that, I was a data scientist in a fintech startup. So my background in data science and data analytics. A few words on Owkin. So at Owkin, we're here to deploy machine learning and artificial intelligence to develop better drugs for patients. We're creating a research ecosystem where we bring together data scientists, researchers, key opinion leaders, pathologists, radiologists, genetic experts, and pharmaceutical company. We all work on patient data that is located within the academic institution firewalls. We don't extract the data, we train our machine learning models remotely, and then we use those machine learning models to extract new medical discoveries. And those are usually biomarkers that we then co-publish, and we also use them to improve clinical trial development.
So thank you, Anna. Maybe just a few words on how did you get to be at Owkin? Maybe a few words on yourself.
Sure. So I met Gilles and Thomas when they were raising their first round of capital back in 2016. And I couldn't invest in them at Boulderton because we had a competitive investment at the time. But I stayed in touch. We worked together over the years, and I decided to join them a few years afterwards.
All right, thank you very much. And now I'm turning to Laurent for your introduction. So Laurent, you're the CEO of Parthenon, and tell us more.
Yeah, great. Yeah, good morning, good afternoon, everyone. Jean-Jacques, thank you for hosting this with the team and Helen, of course. Anna, nice reconnecting. So yes, we are a biotech at Parthenon focused on developing first-in-class therapeutics in the precision oncology space. And the journey to Parthenon for me started off over the past 15 to 20 years, having been in both big pharma and biotech, and developing an appreciation for some of the pain points in our drug discovery and development value chain. And we'll have a chance to talk a little bit more about some of the unprecedented solutions that Parthenon is bringing to the table. But let me just maybe tee up a couple of themes here. So like I mentioned, first and foremost, we're a biotech, meaning that we want to develop unprecedented solutions for patients. And that will be in the form of novel drugs. But in particular, these drugs properly paired to the right patient population. Now, with that in mind, you know, on the one hand, we have developed deep insights into two highly aggressive and deadly cancers over the past several years. One is small cell lung cancer, for which there really haven't been any novel therapeutics identified in the past decades. And then on the other hand, acquired resistance to certain genetically designated cancers such as BRAF mutated melanoma. We've now paired this to a number of novel, unprecedented machine learning algorithms. And these algorithms are really trained by both a combination of patient-derived data as well as preclinical data of all sorts, genomics, transcriptomics, metabolomics, pharmacology, and a few other types of data. So it starts off with a clear problem statement with a high unmet need, lots of data, leveraging machine learning to help structure that data and understand the patterns that are emerging from the data. An output comes out and then back to the lab and then confirming these hypotheses. From my background, so I've been 3-time CEO, twice CSO, and I've seen many projects fail, get into the clinic, go even all the way. And any opportunity that one could identify to so-called bend the probability of success curve going from the lab into the clinic is something that for me is very important. Big picture, I think with machine learning types of approaches, we've gotten to a point of criticality in the sense that while it's not necessarily ready to be deployed in all instances, as long as you have a specific problem statement, there is an opportunity to increase the probability of success and to also decrease cycle times. My background is, like I mentioned, science. It's started off at Pfizer in R&D, and before that, I did a fellowship in genetics and then my training in pharmacology and chemistry. And delighted to continue the conversation. Thanks again, Jean-Jacques.
Well, we are very happy to have both of you for this panel. And I just would like to step back a little bit because I want to understand the whole problem here. I remember I've been in this support to innovation field for quite some time. And I remember 15 years ago, working on various projects, people came to me asking for a lot of money, saying in the next 5-7 years, maybe at most, we will be able to develop a drug in silico. That was 15 years ago. Today, we still see a lot of hype. So is this all hype? Is this reality? Is this being useful? Anna, Laurent, you make use of it every day. How far are we from developing a drug in silico?
Yeah, so Jean-Jacques, maybe let me take a stab at this since we're talking about medicinal chemistry and drug design. So I think, you know, to your point, it comes down to first the quality of the problem. And in this case, asking ourselves, is there an opportunity to identify novel chemical matter, for example, against target X? And there are some recent, well-publicized papers that are very impressive. You know, one in particular is a very nice academic collaboration between Jim Collins and groups at McMaster and other institutions. And basically they were interested in addressing bacterial resistance and specifically trying to address bacterial resistance with novel chemical matter. So the point here is that this is a very high unmet need, very high impact type of problem statement, very precise. Fundamentally, it came down to running virtual screens, which chemists and biologists have done on a computer for many, many years. But I guess ultimately the real question then becomes, can you identify something using machine learning that you wouldn't have otherwise identified? And the question is, in this particular case, like I said, it was a very impressive body of work. And the answer was yes, there was something intriguing. Now, this is one case study, and I apologize to others who have done work in this space, and we probably won't have time to get into it. But the point, though, is that, you know, this is one. We need more of those. And I think it's promising. But ultimately, it's like anything, right? It's stringing together a number of very interesting, unprecedented solutions to a very important problem that will ultimately allow us to say, is this hype, is this reality? I think as practitioners in the field, we do have to be very cautious in the vocabulary and the language that we leverage. I think, you know, all of us, I guess, to some extent or another, we sort of, we're all competing for the gold medal. And sometimes, you know, we get sort of caught up in the need to say, look, you know, we've got something that no one else has done. And so, you know, science gets a little bit of that spotlight that maybe a lot of fields outside of science usually get every day in the newspaper. But I do think that besides, like I mentioned, some very clear progress that have been done using machine learning, there's also a lot of hype and there's a lot of hyperboles. And, you know, I'm not going to point out to any one of them in particular, but I just think that we have to be as good as we are with designing experiments. We have to be that good in communicating what is the reality versus the hype. So to make a long story short, there've been some wins, but I don't think that some of the headlines are necessarily well-grounded in reality at times.
So turning to you, Anna, just to see, I think hype exists everywhere, but you're actually doing it every day as well, using these techniques to facilitate clinical development. So tell us a little bit more about what is reality and what is hype on your side.
I think I have to completely agree with what Laurent was saying. So I think there have been some successes, but they're very minor in terms of impact. So we're not in a place and a point in time where the industry is dramatically changing. I think we're at a point in time where we're seeing a cutting edge technology used with very promising results. I think that regulators are also involved in these decisions and that they are also exploring the use of those technologies in clinical trials. I cannot state today that yes, we can develop a drug in silico or use AI to simulate trials in the next 2 years. That's not possible. It might be possible in the next decades, but not in the very close future. I also think that as part of my role at Owkin, I lead investor relations. The fear of the technology really being helpful, and when I say helpful, I mean really delivering value to pharmaceutical company is key. I think that companies were able to raise in the past based on the technology and the hope for the usability of that technology. I think that today we are expecting to show that it actually is useful and that it creates value for the industry. And so in what we do on an everyday basis, I think we're careful not to oversell whatever we do to pharmaceutical company. I think we're very thoughtful in terms of how we set up the contracts and the milestone and the deliverables and the proof point. And on the other side, for the machine learning models that we build with clinicians and with KOLs, there is a need today to validate them. So if you want to be published in a top-tier scientific journal, you can no longer be published only on your cohort. You need to validate what you have on like one or several independent cohorts. So that's proof that it's no longer about the big headline and the hype. It's actually about what works.
I was on mute. Let's drill into this just a little bit, Anna. It works. You can publish. Tell us more about how it works. What kind of data do you look at? Where does the data reside? How do you use machine learning to help here?
Of course. So we only work on patient data, and we work on multimodal patient data, meaning that we work on clinical outcomes, on pathology, radiology, and genomics. The project that we do are done with clinicians that work in academic institutions. Those are in Europe and in the US, and they are hospitals, foundations, universities, research centers. They all have different infrastructure. So some are on the cloud, other ones use on-premises servers. The data stays there. So whatever infrastructure they're using, we are agnostic, and we've built the connectors to all those type of infrastructures. We set up a project with the clinician. We define internally what are the best algorithms, so what are the best architectures that we have developed in-house to answer the scientific question that is put forward by the clinician. We then remotely send the algorithm to train on the data set that stays local, and we get back the fully trained models. And those fully trained models, obviously it's not a one-way, it's an iterative process. But then those models, we iterate on them a few times to make sure that we get to a result that is robust and that we generalize.
I'm quite intrigued by the idea that the data stays local and that you are able that way to circumvene one of the issues of gathering the data and patient confidentiality. And I suppose this allows you to work faster. Can you give us a concrete example of at the end of the day for some of your clients, the company that work using your help, how much time do they gain? At the end of the day, it's going to be an efficiency question in the clinical trials, isn't it?
Yeah, so maybe I'm just going to take those one at a time. So in terms of how it works, and what's the advantage of not having to extract the data? The advantage is that we are by definition compliant with any type of regulation on data privacy and data security. So we can work with data that is located in Europe, in the US, it doesn't matter, it's the same. Because we're not aggregating the data, we enable collaborative research, meaning that researchers in Europe and in the US can work together without having to pull the data together. And the models are trained simultaneously on distributed data sets at scale. And this is thanks to the federated learning technologies that we have implemented. So this is from a technical perspective, how it works. From a purpose perspective, why having multicentric data is important is because the models are trained on heterogeneous populations. Those populations are different in terms of patient data, so the genetics of the patients are different, but also the treatments that they have been following are different. And finally, the data itself is different because the way data is recorded in France in a specific hospital versus how the data is recorded in New York in another specific hospital, it's different. And being able to train model in this heterogeneous, multicentric setting, it's important to have them perform well and generalize to a larger population. And why is that important? That's essential for pharmaceutical companies if they want to use the models to analyze and design clinical trials. So to go back to link all this together, the federated technology enables us to produce models that are exposed to very large panel of patient and data. And that then gives us the opportunity to have models that will be generalizable to the specific population that the pharmaceutical companies are using for their clinical trials.
All right. Just a quick mention to all of the audience. We already have a few questions that have come through the Q&A function. Please don't hesitate to put questions to our speakers, again, by writing down a question in the Q&A. And we will address the one we have very shortly. Laurent, tell us a little bit more about data also, because you mentioned how in the works that you do, you use many types of source data to do your work. Tell us more about that and how you, we have heard a little bit about the Jim Cohen's work at MIT. What Parthenon does on the biology side is quite different from just medchem and innovative use of machine learning to explore medchem.
Yeah, that's correct, Jean-Jacques. So let me maybe take a quick step back here. If we break down, as you know, the value chain across R&D, obviously beyond the separation of chemistry and biology, there's also the opportunity to understand disease mechanisms. And so we at Parthenon are laser focused on understanding the root causes of, on the one hand, acquired resistance in specific cancers, and on the other hand, also understanding how do we best simulate the plasticity of cancer cells so that we can reprogram them. So that is in essence what we do at Parthenon, which is, again, on the one hand, our focus is on reprogramming cancer cells that have evaded the natural surveillance mechanisms inside the body, so that we can now better understand how to intervene therapeutically. So in the case of small cell lung cancer, as I briefly mentioned, we have leveraged a number of different lines of data, both preclinical data as well as patient-derived data, in order for us to better capture how does the cancer signal and evade the different mechanisms of defense that most of us have and that work well all the time. Now, the reason for that is because in order to be able to break the signaling code, you have to be able to understand and speak the alphabet of the cancer. And so by mapping out the different signaling nodes inside a cancer, we're now able to formulate a therapeutically based strategy that would then allow us to develop new molecules against sometimes novel targets that no one else has ever developed a drug against. So that's one objective. The other objective at Parthenon is also to better co-target the disease that we're trying to solve for. And by co-targeting, what I mean is simply to say one of two things. Very often in oncology, polypharmacology, combination of drugs is used to really knock out the cancer. And so very often what we've realized is that there's no good rationale right now. There's no good rationale approach to combining drug A and B. So that's one of our objective, is to solve for that. The other is, as many of you know, the right context for a single agent is also very important, meaning what are the co-targets that make that particular drug more powerful or perhaps less powerful. And so this idea of co-targeting is especially important, and it doesn't imply that you're co-targeting with two or more different agents. So we have developed algorithms that, on the one hand, using probabilistic Boolean network, have allowed us to map the signaling nodes within a specific cancer, in this case, small cell lung cancer. And that particular type of approach can now be transposed to other cancers. And then on the other hand, we've developed deep neural networks to actually help us understand how do you co-target a particular cancer. And in our case, we originally developed that algorithm to go after BRAF mutated melanoma in the context of standard of care. So again, all of these algorithms, all these technologies, unprecedented, but they always had a problem in mind, and they were always developed with a problem and then a projected solution. So maybe let me pause here and see, Jean-Jacques, if you wanted to go on to something else.
What I find very interesting in what you just described is both modelization using probabilistic models and machine learning to explore the pharmacologic application there. Do you have to go in vivo anytime? Check what you're doing? Because one machine talking to the other, you could get strange results, maybe.
Right. So like everything, you know, there's a process and a workflow that's been put in place. And as I mentioned, you know, it starts off with a lot of data. But where machine learning is helpful is you don't need 100% of the theoretical amount of data that is required to understand a situation. That's where machine learning can help you recognize patterns and help fill in the blanks and connect the dots without necessarily having all the dots in between two dots. So lots of data, lots of biology. Then there is a, let's call it a mathematical step that comes after that in order to make sense of all of this and allowing us to model, simulate, and understand, you know, if I disrupt this signaling node, what is the consequence of the cell? But the third step is absolutely essential, and I think this is what distinguishes some companies from others, is that you gotta have a biological validation step. And by that, what it means more granularly is that you've got the typical in vitro assays, in vivo assays, and that's when you do really drug discovery, is, you know, you apply your chemistry, your biology, and you run all the experiments needed to file an IND in order to make sure that what you got out of the algorithms actually is true.
We have one question here. Let me throw it out to you just now, and reminding all of our listeners to not hesitate to ask questions for the Q&A. So these new compounds that are found from these machine learning algorithms, how diverse are they? Are they really innovative drugs? Can machines invent?
Yeah, so you know, one way for me to interpret this is twofold. Well, you know, this whole topic of machine learning, I find it fascinating because at some level, you know, it gets us into philosophy very quickly. You know, what is obvious, what is not obvious? I mean, behind these questions, that's really what we're asking. And one could argue that there is nothing obvious, so therefore it's all patentable, and therefore it's all novel to some degree, right? So that's one approach you can take. But just to be a little bit more sort of down to earth here, the idea of unexpected, sort of unpredictable, can be maybe of two kinds. One is you have an existing molecule that's been discovered to do something, and now you have that same molecule that's now being discovered to do something new that no one else had ever discovered before. And that is in some ways what you find in this famous paper now that we've talked about from Jim Collins at MIT, is that they found some existing molecules that do something new. So that's pretty cool, right? I mean, that's like sort of repositioning, repurposing, you know, to some degree. And relying simply on human intuition is just sometimes too tough, right? But I'm not suggesting that humans have not been able to do that. There are many, many examples of that. So the other way to answer this point or to comment on it is to say, okay, I've got a molecule, and now I've identified quote unquote novel molecules, and how different are the molecules from one another? And this is where, you know, the debate becomes a little bit sort of in the eye of the beholder, because it's tricky, right? Is it one atom? Is it two atoms? Is it a full ring structure that is now described with a machine learning approach? Could that have been done using computational tools that don't rely on neural networks? So I would say that I haven't yet seen a clear example of something that, at least as, you know, from a purely, let's call it intuitive perspective, is brand new, right? That machine learning, we can say, wow, you know what, no one else could have ever thought about this molecule. But you know what, I don't make any claims to know the full extent of the literature. I'm sure as I'm saying this, there are going to be dozens of examples that contradict me. But overall, I think that's fundamentally the key question is, can we have multiple examples where non-classical virtual screen computational chemistry approaches have been outcompeted by neural networks to identify molecules that on paper look very different? And I'm not just talking a couple of atoms here. I'm talking brand new chemical structure. The tricky part ultimately, and I'll just wrap up, Jean-Jacques, is that you always want to link those structures to function. And that's the part where simply having for example, a target that you're running a virtual screen against, and then saying, I found new chemical matter, that's not good enough. Ultimately, that is not a drug or even a drug candidate. You really need to have function. You need to have biology that ultimately comes and demonstrates that even if you have a brand new structure, can this become a drug? And I don't believe I've seen enough of those examples just yet. But, you know, I'm sure people are working on this, you know.
So I want to make sure I get to a lot of the questions that we have here. So very quickly, because people are wondering a little bit about how much effort it takes to do all this, can you just, both of you, tell to our listeners how big your companies are? How many people do you have in Parthenon, Laurent?
Yeah, we've got about 2,000.
And how about you, Anna? How many people in Owkin?
100. And we have 50 data scientists.
So you are both rather small startups, if I may say. I mean, quite reasonable and doing very well. But how do you compete with the large tech companies? Are those going to become CROs for the pharmaceutical industry? Do they have the knowledge of the clinical development setting that is necessary for them to become competitors in that field? Who is your competitor, Anna?
I think it's a great question, and I think maybe I can start, Laurent, if that's okay.
Please.
And to build on what you were saying, I think the key is the interpretation of the results that the AI models provide. I think that's the biggest differentiation between biotech startups and large AI companies. I think there is a need for cutting edge AI, but there is even a bigger need for interpretation and biology behind what you find. And we are in a field that AI black box models won't work, won't be acceptable. So I think that we want to be able to validate any prediction that we get out of our models. And we want to validate those with the clinicians that we work with. So that maybe I can talk a little bit more about our infrastructure as well. But at Owkin, the models that we build are the technologies and the type of neural networks that we use depend on the data modality underneath. But at the end of it, we always have interpretation features, which we are putting a lot of efforts on developing. And those are required by the researchers to understand in non-small cell lung cancer, in breast cancer, in any oncology areas that we work in, to really understand why patients are responding or not responding in a certain way, and why the disease is evolving in a certain way within specific subgroups of patients. And the AI infrastructure is built in a way that we can identify the signal, the features within the data that were responsible for the prediction of the models. And then we go as deep as we can into those, on as many as we can, to validate their biological meaning. So are those specific cells, or are those specific areas of the tissues or genes involved in the mechanism of action of the compound, or in the specific pathways? So we have to validate and answer all of those questions in order to come up with a publication, but also to come up in front of a pharmaceutical company and say, you can use this model to actually better understand your clinical trial and better understand efficiency in your clinical trial. Efficiency is what measures the the effect of the investigated drugs. That's what at the end is important for the pharmaceutical company. And so to maybe to come back to your point, the value here is not really on being able to build the best AI in the world. The value here is to build an AI that is like deeply rooted in biology. And I think that's the key differentiator that we have at Owkin. So obviously we're not Google, and we're never going to be Google, but also we're not Novartis. So we're kind of like in the middle. We are a machine learning and AI company that has developed federated learning technologies to enable the distributed training, and that has a very strong expertise in biomedical AI.
So
Jean-Jacques, may I just take a second here to follow up on this? So, I mean, I couldn't agree more with Anna. I think she said it really well, and maybe just to double click on a few things here. So, if I may, you know, perhaps there's an opportunity here to level set and recontextualize a little bit the position of AI and machine learning. And what I mean by that, the vision here, and it's a very tactical vision to some degree, is we don't think that you want to start with AI ML at the very top of your value proposition. Our vision, though, is that it is difficult, especially for me, to imagine 10 years from now, any biotech not doing AI ML as part of its everyday operations. It's kind of like saying, you know, don't do biochemistry or do biochemistry. At least from the R&D perspective, it's difficult in light of the vast amount of data that's out there and all the unknowns and the limitations of human intuition to basically go out and build a new company that doesn't have the best possible mathematicians and machine learning experts for the future. So I personally think that over the long arc, we're on a irreversible trajectory that any competitive biotech and anybody who's listening who wants to build a biotech, who wants to develop novel medicines, I would strongly urge you to ask yourself, can I afford not to be operating in the machine learning space? And obviously what I've just said, you know, there's a lot behind that in terms of discussions, and I'm happy to have that maybe afterwards during the breakout sessions. But I also think that why I wanted to mention this is because the conversation is not about is AI ML impactful to biotech. It's more how does one accelerate the discovery or discoveries in general? How does one get into the clinic? How does one shorten cycle times? How does one increase probability of success? And if I knew that there was a 1% chance that machine learning could do it, then why would I not do it, right? So I think we all have a responsibility when I think about the sector to do all we can to deploy all the best technologies that can get us into the clinic and onto the market as quickly as possible. And I just wanted to sorry, just because I see a little question, there's a little screen there and I see a bunch of good questions there. And just in terms of the positioning of tech companies in the biotech ecosystem, I think it comes down to business models. In our case, you know, we're building a fully integrated biotech that goes from idea generation, ultimately all the way to clinical proof of concept and approval. But it doesn't mean that that business model works for everybody else. Others might want to be service providers or strategic partners of other companies. So again, AI ML is more of a mindset and a set of tools that fit into a bigger context.
So thank you for this. We're coming to the end, but I just would like to ask you both a somewhat personal question, but that links back to our listeners. Laurent, you are in the Boston area on the East Coast of the US. Anna, you are in New York, if I'm not mistaken. Can you build a company that has the appropriate technologies in France? Or do all our listeners have to come to the US?
Maybe I can take that to start. I think that for us, the company is based in New York, but we also have a French R&D. Most of the R&D is actually located in France. So I think that in both Europe and in the US, you can attract very good talent in terms of machine learning and AI. You have specific countries where you have really good schools and universities specialized in machine learning. So for instance, you have France, but you obviously have Canada with the Mila Institute. So those are kind of everywhere now in the US, in Boston with the MIT, obviously, but they, I don't think it's difficult is not to attract the talent. I think the difficulty is to locate yourself in the environment where you're going to have kind of the full stack. So where you're going to have the researchers, the machine learning expert, and the clinical trial, so the pharmaceutical expert as well. And I think that there, maybe the US, there is an advantage in being located there.
A 10-second comment, Laurent.
Yeah, I think, you know, maybe same idea expressed perhaps in different words. You know, I think obviously to build a great biotech, you got to have great people. And there are many places, cities, countries where there are great people. Having said that, you also need a platform that allows you to integrate all these great people. And, you know, in the case of Boston, you know, there is an ecosystem in the sense that you've got R&D talent, you've got investors, you've got banks, you've got pharmas. And so there is something about that ecosystem. But at the same time, you can recruit talent from many different locations as long as you're able to integrate them in a way that helps you move forward with a business. So I would say that it's absolutely fine to create a multinational biotech from day one with teams all across the country. But at the same time, you do need a team of decision makers that is well plugged into a efficient ecosystem. And I think as the stock market in the US has been telling us over the past few months, US biotech and access to cash is quite good here. And so that's, I think, a consideration, you know, for anybody who wants to get into this space. You're on mute.
We would really appreciate you to fill in the questionnaire as you leave this webinar. The networking session will start in just a few minutes at the link which is in the chat. And we look forward to sharing with you and meeting you again at our next event, which will take place on October 1st. Announcements to follow. We're focused on the markets of oncology. Thank you very much, and we look forward to seeing you soon again. Bye-bye.